right. Hi, everybody. It's 11 o'clock here in Minnesota, so we're going to go ahead and get started. Thank you, everyone, so much for joining our webinar today. My name is Jackie Carville, and I'm the Marketing Director here at ImmunoChemistry Technologies. I will be moderating this webinar. You may have noticed that your phone has been muted. However, we do encourage you to ask questions along the way. Simply type your question into the chat dialog at the right and select Send to Host. If you have any technical difficulties during the webinar, please feel free to send your chat to me and I will try my best to get you up and running. We will be posting the video recording of this webinar later this afternoon, so do look out for an email from me after the webinar with a link to that recording. Now I'd like to go ahead and introduce our speaker, Dr. Christy Strandberg. Now, Christy is a staff scientist here at ICT where she develops new products, completes service projects for customers, and is a fantastic consultant for all your technical support needs. Today she will be going over several of our fluorescent cellular assays, including a discussion of apoptosis, pyroptosis, and autophagy. And with that, I'm going to go ahead and turn it over to Christy.
Well, thanks, Jackie. Now, before I dive into today's webinar topic, I want to take a quick moment to tell our listeners a little bit about ImmunoChemistry Technologies. We are located in Bloomington, Minnesota, which is a suburb just south of Minneapolis. ICT just celebrated its 22nd anniversary this past September, so we've been in the immunoassay business for quite some time. We are a privately owned company that was incorporated back in 1994. With our expertise in immunoassays and ELISA development, we do offer a variety of immunoassay-related services, including assay design and development, antibody purification and conjugation, lyophilization, and so on. Our scientists have years of experience with protein chemistry and ELISA optimization, so we can help you develop reliable, sensitive, and specific immunoassays. We can also scale up and manufacture an assay for internal use for clients. If you're in need of a service project, feel free to reach out and get in touch with us. We'd be more than happy to discuss your project. A few years ago, we expanded our company to offer lines of consumable reagents. Now, this includes our ELISA Solutions, and this is items such as coating buffers, blockers, diluents, conjugate stabilizers, and such. We also offer a line of products tailored for detecting cell health status. Before we move on to discussing our solutions for detecting cell death pathways, I want to mention our ELISA Solutions. This product line includes all the components you need to build a better ELISA. ICT's coating buffers, blockers, sample and assay diluents, conjugate stabilizers, and wash buffer all work together to minimize the buildup of unwanted proteins to generate a very clean signal. These products work together to address issues commonly encountered during ELISA development, such as matrix effect or interference. Our products are also designed to promote long-term stability and improve shelf life. The ultimate goal is to help you develop optimized ELISAs that have a high specific signal and low background noise. That's all I'll mention today about our ELISA products, but if you do have questions about this product line, feel free to chat them in and I can get a response to you after the webinar. As the webinar title implies, today's presentation will focus on ICT's product lines for detecting different cell death pathways. Our cell death detection products include a variety of fluorescent whole cell assays for detecting intracellular apoptosis, necrosis, cell-mediated cytotoxicity, pyroptosis, autophagy, and much more. I would like to emphasize that these kits are not ELISAs. Instead, they use cell-permeant fluorescent reagents to analyze whole intact living cells. Here's a short agenda of today's webinar topic. I'll start by giving a little bit of background on cell death, the roles of enzymes called Caspases in the forms of programmed cell death such as apoptosis and pyroptosis. I will also introduce our FLICA product line for detecting active Caspases. From there, I will briefly introduce our audience to some of the assays we offer for detecting various cell death pathways, including our new kit for detecting autophagy. Cell death is typically categorized into non-programmed or programmed cell death pathways. Necrosis is a form of cell death that does not involve activation of a programmed cell death pathway. The term necrosis has come to have several meanings. Pathologists use the term necrosis to designate the presence of dead tissues or cells. Oftentimes, the term is used to describe accidental cell death that is believed to have occurred in a manner other than a programmed cell death process. However, it should be noted that overall, at its most basic level, necrosis is the sum of changes that have occurred in cells after they've died, regardless of the prelethal process. Necrosis therefore refers to the morphological state seen after a cell has died and has reached equilibrium with its surroundings. In the absence of phagocytosis, which I'll point out is often the case in cell culture-based experiments, apoptotic bodies may lose their membrane integrity and proceed to a secondary or apoptotic necrosis. Programmed cell death is traditionally broken down into two categories, non-inflammatory and inflammatory. Apoptosis and autophagy are non-inflammatory cell death pathways, while oncosis and pyroptosis are considered inflammatory pathways. All are important for maintaining homeostasis. Examples include the non-inflammatory normal cell turnover seen during apoptosis, as well as the inflammatory response to infections seen during pyroptosis. Sorry, 1 second, trying to advance the slide. There we go. I want to take a minute to further compare and contrast these different cell death pathways. Apoptosis involves cellular dismantling that occurs in a manner that avoids eliciting inflammation. Apoptosis features activation of initiator caspases that activate effector caspases, which then go on to cleave cellular substrates. Apoptotic cells demonstrate cytoplasmic and nuclear condensation, DNA damage, formation of apoptotic bodies, maintenance of an intact plasma membrane, and exposure of surface molecules targeting the intact cell corpses for phagocytosis. Autophagy features degradation of cellular components within the intact dying cell in autophagic vacuoles. The morphological characteristics of autophagy include vacuolization, degradation of cytoplasmic contents, slight chromatin condensation. Autophagic cells can also be taken up by phagocytosis. Now, oncosis is an inflammatory cell death pathway that's accompanied by cellular and organelle swelling and membrane breakdown. This membrane breakdown leads to the eventual release of inflammatory cellular contents. And last but not least, pyroptosis also leads to the release of inflammatory contents, but is a cell death pathway that is mediated by the activation of caspase 1. Caspase 1 also activates the pro-inflammatory cytokines IL-1 beta and IL-18 to their active inflammatory forms. So, with that in mind, this pathway is obviously inherently pro-inflammatory. Pyroptosis also features cell lysis and the release of inflammatory cellular contents, but I would like to mention, undoubtedly, other pathways most likely exist and have yet to be discovered. I want to discuss apoptosis in greater detail. Apoptosis is activated through one of two different pathways, either the intrinsic pathway or the extrinsic pathway. Both pathways involve activation of initiator and effector caspases. The intrinsic pathway is activated following cellular stress, which results in mitochondrial perturbation and the release of cytochrome C. Once released, cytochrome C binds to the apoptotic protease activating factor 1, or APAF1, triggering the formation of the apoptosome complex, resulting in activation of initiator caspase 9. Once activated, initiator caspase 9 then processes and activates the effector caspases 3, 6, and 7. Now, the extrinsic pathway is initiated following activation of death receptors of the TNF superfamily. The trimerized receptor ligand complex recruits procaspase 8 to the death-induced signaling complex via Fas-associated death domain, leading to the rapid activation of caspase 8. Once activated, initiator caspase 8 then processes and activates effector caspases 3, 6, and 7, which are responsible for the phenotypic changes associated with apoptosis. So what exactly are caspases? Caspases are a family of proteases that serve as critical mediators in apoptosis and pyroptosis. They are named caspases due to their specific protease activity. A cysteine in the enzyme's active site nucleophilically attacks and cleaves the target protein at the C-terminal of an aspartic acid amino acid, which is why the enzymes are called cysteine-dependent aspartate-directed proteases. Caspase substrates are cleaved after usually a tetrapeptide sequence containing aspartic acid in the P1 position. Mammalian caspases are traditionally broken down into three functional groups: initiator caspases, executioner or effector caspases, and inflammatory caspases. The apoptotic initiator caspase group includes caspases 2, 8, and 9, which are found in humans and mice, and caspase 10, which I believe is found in humans only, As their name implies, these initiator caspases are involved in the initiation of the caspase cascade. Apoptotic executioner or effector caspases include caspases 3, 6, and 7, which are found in both humans and mice. These effector caspases are involved in apoptosis and function to dismantle the cell by cleaving various cellular substrates. The inflammatory caspases include caspase 1, which is found in humans and mice, caspases 4 and 5, which are found in humans, caspase 11, which is found in mice, and caspase 12, which is found in mice and some humans. I want to delve a little deeper into caspase 1 and pyroptosis. Pyroptosis, as I mentioned before, is a highly inflammatory form of programmed cell death that occurs upon situations such as infection and likely plays a role in the host antimicrobial response to pathogens. Caspase 1 is activated during pyroptosis by a supramolecular complex called the inflammasome. Unlike apoptosis, cell death by pyroptosis results in plasma membrane rupture and the subsequent release of intracellular contents. This contrasts significantly from apoptosis, where the cellular contents are packaged into apoptotic bodies, ultimately to be taken up by phagocytic cells in a non-inflammatory manner. The morphological differences between apoptosis and pyroptosis are highlighted nicely in the image shown on this slide. Note the intact membrane blebbing occurring on the apoptotic cell compared to the ruptured membrane of the pyroptotic cell. So this slide depicts caspase 1 activation by the NLRP3 inflammasome. Caspase 1 is cleaved and activated in response to multiple stimuli. Activated caspase 1 initiates pyroptosis. Activation of caspase 1 leads to the rapid formation of membrane pores that allow for the dissipation of cellular ionic gradients, thus causing swelling and osmotic lysis. Proforms of IL-1 beta and IL-18 are also processed by caspase 1 and released. Caspase 1 activity results in the cleavage of chromosomal DNA, but the fragmentation, I do want to point out, this fragmentation differs from that seen during apoptosis. Nuclear condensation occurs, but nuclear integrity is maintained. NLRP3 activation usually requires two signals. The first signal begins with the recognition of a pathogen-associated molecular pattern by toll-like receptors, such as the recognition of LPS by toll-like receptor 4. Then, through an interaction with the adapter protein MyD88, we see triggering an activation that leads to, this triggers activation this activates the transcription factor NF-kappa B. Once activated, NF-kappa B is translocated to the nucleus, where it leads to the synthesis of inactive pro-inflammatory cytokine pro-IL-1 beta. Another potent pro-inflammatory cytokine precursor, pro-IL-18, is constitutively expressed. However, its expression is increased after cellular activation. The second signal is triggered by an ionic perturbation in the cell, such as an influx. Second signal, such as ATP, will lead to ionic perturbation in the cell, such as efflux of potassium. This subsequently results in assembly of the NLRP3 inflammasome, caspase 1 activation, IL-1 beta, and IL-18 secretion. Now I would like to switch gears and introduce ICT's line of FLICA products, which are designed to detect caspase activity. FLICA stands for Fluorescent-Labeled Inhibitor of Caspases. FLICA are cell-permeant probes that are able to traverse the hydrophilic lipid bilayer of cells because of their low molecular weight, absence of any charged structure, and their overall hydrophobic character. All FLICA products contain the same general structure, a unique peptide sequence that competes for caspase binding that is flanked by a fluorescent reporter tag, such as carboxyfluorescein, as well as a fluoromethyl ketone moiety that is responsible for irreversibly binding to the active caspase. The upper image on this slide shows the contents of our FAM FLICA kit, and the lower image shows THP1 cells that have been duly stained with our polycaspase FLICA probe, as well as hooks to stain DNA. In the fluorescence microscopy image, only the middle cell is apoptotic, as seen by the green cellular staining indicating caspase activity. The middle cell also has many bright blue spots indicating that the cell is beginning to die. The cell in the upper right corner is necrotic. We see no green, and we see a scattered blue staining. The cell in the lower left corner is alive and not apoptotic, which is evidenced by a lack of green staining, as well as a concentrated blue stain for the DNA. Before I move on, I want to give our listeners a quick high-level explanation of how FLICA works. When the caspase is in its inactive zymogen form, the enzyme's catalytic sites are blocked. Once processed to its active form, these catalytic sites become exposed. The FMK, that fluoromethyl ketone moiety, on the FLICA probe is then able to form a covalent bond with the reactive catalytic sites on an active caspase enzyme. This enables the FLICA probes to be retained inside the cell despite any subsequent wash steps. It's important to note that FLICA is not cleaved by the caspase. Once the FMK reactive groups on the FLICA probe form covalent bonds with two of the catalytic cysteine groups in each of the two caspase reactive sites, that particular caspase enzyme is rendered essentially inactive, meaning it can no longer cleave any substrates. Here are some sample flow cytometry results showing our apoptosis detection kits. Here I'm showing you the data on the left shows our sulfurrhodamine DEVD FLICA probe. This DEVD sequence preferentially targets caspases 3 and 7. On the right is our 660 far red VAD FLICA. VAD is a sequence recognized by many caspases, and thus we consider this to be our polycaspase probe. As I mentioned earlier, our FLICA probes contain the preferred amino acid binding sequence for the caspase of interest. The histograms shown on the slide depict the results from experiments where Jurkat cells were induced to become apoptotic by a 4-hour exposure to staurosporin. After this induction period, the cells were incubated with FLICA for 60 minutes, during which time the FLICA probe passively diffuses across the cell membrane, where it can then bind to active caspases. Any unbound FLICA is removed during subsequent wash steps. After the wash steps have been performed, the cells are ready for analysis. So as you can see in the data I'm showing you here, in both instances, the caspase 3-7 dye or the polycaspase dye, treatment with staurosporin induced over 90% of the cell population to initiate apoptosis. If you compare the negative control non-induced population to our staurosporin apoptosis-induced populations, you can see a nice shift in fluorescence signal. Our kits are also compatible with fluorescence plate reader and fluorescent microscopy instrumentation. So here's some representative data showing those instances. The plate reader data shows Jurkat cells that were non-induced or were induced with staurosporin for 4 hours and then stained with ICT's FAM-VAD polycaspase FLICA probe. The microscopy results depict staurosporin-induced cells that were stained with our far-red 660 DEVD probe, which, as I mentioned on the last slide, detects caspases 3 and 7. Let's not forget about caspase 1. We have FLICA products for detecting caspase 1 activity as well. So here's some data that depicts using flow cytometry to detect caspase 1 activity. ICT's caspase 1 reagent, this is a FAM-VAD FMK FLICA probe, was used to monitor caspase 1 induction response in Jurkat cells that were treated with nigiricin for various periods of time. A common cell pool was spiked with FLICA and then divided into separate treatment groups. Starting with the 24-hour samples and working backwards, 10 micromolar nigiricin was added to cells and the samples were incubated at 37 degrees throughout the induction process. Following their respective treatment exposure periods, the cells were washed and analyzed on a flow cytometer. The amount of caspase 1 activity detected directly correlates to the duration of the exposure period. What I mean is the longer the cells were exposed to nigiricin, the larger the proportion of caspase 1 positive cells within that sample. This slide is showing some fluorescence microscopy results obtained from colorectal epithelial cells that were infected with the Salmonella strain constitutively expressing M. cherry, which stains red. After 9 hours of infection, the cells were then incubated with ICT's green FAM-YVAD FLICA probe for 1 hour. Afterwards, the cells were washed and then fixed and then evaluated on a confocal microscope. This image reveals a cell that's extruding. This is an infected cell that's extruding. You can see that many red M. cherry labeled salmonella are visible within the cell. The infected cells are undergoing pyroptosis, as evidenced by the positive staining for active caspase 1, visible as the increased green fluorescence signal, compared to background levels of fluorescence in the surrounding caspase 1 negative cells. Now, before I move on to discussing some of our combination kits, I want to mention our Magic Red product, which also detects caspase 3 and 7 activity. Unlike the FLICA probes, which inhibit the enzyme's activity by covalently binding to the caspase active site, Magic Red serves as a caspase substrate and can detect caspase activity without inhibiting the caspase. Magic Red is a membrane-permeant probe that's dye-substituted with 2 DVD amino acid target sequences. As I said before, DVD is a preferred target sequence for caspases 3 and 7. When Magic Red is in this dye-substituted form, the fluorescence signal is quenched. However, since this molecule serves as a substrate, as active caspases 3 and 7 cleave off the DVD groups, the probe shows an increase in fluorescence activity. Although not the focus of today's webinar, I do also want to mention that ICT carries a line of Magic Red probes for detecting cathepsin activity. Our cathepsin detection products recently featured in a different webinar and won't be discussed here. If you're interested in viewing some of our older webinars, just go to the archive section on our webinars page. Now I'd like to discuss some of our combination kits. The Necrosis versus Apoptosis kit combines our FLICA technology with 7AAD to allow for an enhanced understanding of cell death by simultaneously identifying necrosis and apoptosis. 7AAD is short for 7-aminoactinomycin D. This is a red fluorescing live dead stain. It can enter cells that have compromised cell membranes and will then go on and bind to DNA. It will not enter living cells that have intact cell membranes. So here at ICT, we recognize that there's a considerable demand for research tools that can allow scientists to determine how cells are dying. This is very, very prevalent when investigating novel drug treatments. It's absolutely critical to evaluate biological or chemical compounds for unintended cytotoxic effects on cell cultures. Beyond being able to assess cell death as an experimental outcome, it's extremely important to have some reassurance that your indicator cell population is healthy prior to initiating any experimental protocols. With that in mind, ICT created this necrosis versus apoptosis detection kit. And I'll show you some data later, but improper cell handling and cell culture maintenance can lead to unhealthy cell cultures, thus potentially introducing artifacts or leading to difficult to interpret experimental results. A common mistake we hear of is labs that maintain cell cultures at an inappropriate cell density. Long-term maintenance at too low or too high a density can have detrimental effects. Like I said, I'll show you some data supporting that in a little bit. The combination of FLICA with 7AAD allows for simultaneous detection and quantification of 4 distinct cell populations. Any live, non-apoptotic cells will remain unstained as they do not contain active caspases and have intact cell membranes, which exclude 7AAD. Cells that have entered early apoptosis will fluoresce green with our FLICA product as their caspases have become activated, but they will not fluoresce red as 7AAD is still excluded by the intact cell membrane. Cells that have entered late apoptosis will fluoresce green and red due to the presence of active caspases and loss of cell membrane integrity. Dead cells will fluoresce red only as they do not contain active caspases and have lost their cell membrane integrity. So our necrosis versus apoptosis detection kit is ideal for monitoring cells by flow cytometry. When you plot the FLICA using the FL1 channel and the 7AD using the FL3 channel, you can easily identify these 4 cell populations as they show up in separate quadrants. The lower left quadrant will house the live unstained cells. And the lower right quadrant will contain the green staining early apoptotic cells. The upper right section will contain the green and red labeled apoptotic cells, and the upper left section will house the dead cells that have stained red only. So the data I'm showing you here is comparing mock-treated cells versus staurosporin-treated Jurkat cells. These have been duly stained with FLICA and 7AAD. And what we see is that the majority of the mock-treated cells fall into the live unstained quadrant, while very few of the induced cells remain in that quadrant. Just over 46% of the staurosporin-treated cells have entered into early apoptosis, and over 50% are in late apoptosis. As I mentioned earlier, improper culturing techniques can lead to unhealthy cell populations. Here, I have intentionally allowed Jurkat cells to become overgrown prior to staining with our necrosis apoptosis kit. Cells that were maintained at concentrations ranging from about 1 × 10⁵ cells per mil to 6 × 10⁵ cells per mil appear to be healthy, as most of the cells are located in that lower left quadrant. As our cultures were allowed to reach concentrations of 1 times 10 to the 6th or greater, we see a pronounced increase in the number of cells entering early apoptosis, as evident by an increase in the FL1 signal. Again, these are just cells left growing in culture. I've not added any sort of treatments to them other than letting them become overgrown. Our necrosis versus apoptosis kit is also compatible with fluorescence microscopy. This slide shows images of Jurkat cells that were treated with staurosporin for 4 hours. Here you can see early apoptotic cells that stain green only, cells in late apoptosis that stain green and red, and then dead cells that stain red only. I don't generally recommend this apoptosis necrosis kit for fluorescence plate reader. Certainly it's possible to use it, but it just becomes more complicated measuring 2 different wavelengths of the 2 different probes simultaneously. Now I want to switch gears again and discuss ICT's combination kits for detecting cytotoxicity. Before I get too far ahead of myself, let me first explain exactly what I'm referring to when I mention cytotoxicity. Cytotoxicity in general is something that kills cells. Cytotoxicity or cytolytic activity is an important process for eliminating intracellular pathogens such as cancer and cancer cells, and has both innate and adaptive effector cells. So what I'm talking about for these next few slides is cell-mediated cytotoxicity. And this occurs when cells of the immune system, hereafter called effector cells, act on another cell, which from now on will be called the target cell. Often the result of this interaction is to kill the target cell. In the picture I'm showing you here, what we see are 2 natural killer cells. These are showing up as the yellowish-orange cells on either end. They're attacking a red cancer cell that's in the middle. The natural killer cells are the effector cells. They are having some sort of impact on another cell. That cell is the red cancer cell, which we're calling the target cell. So when I'm discussing our cytotoxicity kits, this is the kind of cell-mediated interaction that we're interested in. ICT offers 2 cytotoxicity kits, and both of these kits build on our FLICA and our SR FLICA and 7AAD platform. So we offer a basic cytotoxicity kit that is comprised of a CFSE and 7AAD, as well as a total cytotoxicity and apoptosis kit, which includes a CFSE and 7AAD, but then also adds in our sulfur-rhodamine polycaspase FLICA probe. Both the basic and the advanced cytotoxicity kits were designed to work with target and effector cells. So let's start by going through the more simple explanation presented by the basic kit. The basic cytotoxicity kit distinguishes live target cells from necrotic cells without interference from the effector cells, and allows for an easy quantification of cytolytic killer cell activity without the need for lysing cells, waiting for enzyme release, or using radioisotopes. The basic kit measures necrotic and membrane-compromised cells in a mixed population of effector and target cells when grown in culture. So to use this kit, what you need to first do is isolate your target cell population as well as your effector cell population. The effector cell population, such as natural killer cells or cytotoxic T lymphocytes, will remain unstained. The target cell population will be stained with CFSE, which allows us to gate on the target cells later. The target and effector populations are then mixed together. Cytolytic activity is then detected by adding the live dead stain 7AAD. Cells are then analyzed by flow cytometry. During our analysis, we create a histogram that plots the CFSE on the FL1 channel and 7AAD on the FL3 channel to reveal live unstained effector cells in the lower left quadrant. The dead effector cells will stain red and show up in the upper left quadrant. Likewise, the CFSE stained target cells are also revealed. The live target cells stain green and show up in the lower right quadrant, while the dead target cells stain green and red and show up in the upper quadrant. So let's move on to the total cytotoxicity and apoptosis kit. Like the basic cytotoxicity kit, the total kit also contains CFSE and 7AAD. The total kit differs from the basic kit in that it also includes our sulfur-rhodamine polycaspase FLICA probe. The addition of FLICA allows for a more accurate assessment of the cytolytic activity by concurrently labeling target cells that are apoptotic. Like with the basic kit, only the target cells are labeled with CFSE, so the effector cells can still be distinguished from the target cells. The effector and target populations can be resolved by plotting FL1 by side scatter. If you take a look at our plot number 1 on the slide. Next, the live and dead cell populations of both target and effector cells can be seen by plotting CFSC on FL1 channel versus 7AD on the FL3 channel. It's shown in our second plot on the slide. The green cell populations are the target cells, and the red stained populations are the dead cells. From there, we can gate on the CFSC stained target cells and then plot FL2 to show the orangey red staining of our sulfur-rhodamine FLICA versus the FL3 red staining of the 7AAD live dead stain. When we look at these 4 quadrants, as shown in plot 3, we see that the healthy live target cells show up in the lower left quadrant and stain green only. Live cells that have entered early apoptosis will be found in the lower right quadrant. During early apoptosis, caspases are active, but the membranes are still intact, as I mentioned before. Now, without the presence of our FLICA probe, these cells would have excluded the 7AAD vital dye, and thus would have been inaccurately found in the live healthy quadrant. The addition of FLICA also allows for discerning the cells in late apoptosis from the dead or necrotic cells. Late apoptotic cells would still have active caspases, as well as leaky cell membranes, and would be detected in the upper right quadrant. Lastly, dead or necrotic cells can be found in the upper left quadrant. So overall, our basic and total cytotoxicity kits make for an easy, safe, and sensitive way to measure cell-mediated cytotoxicity. Now, if your cytotoxicity needs are a bit more general, i.e. you're more interested in getting an idea of the overall health or viability of cell population, you might wish to consider our Calcein AM-based kits. Calcein AM is a versatile membrane-permeant cell viability marker. Please note that Calcein AM can be used as a general viability stain and is not meant to specifically detect cell-mediated cytotoxicity in target cells. Calcein AM is designed as more of a general cell viability stain or a live dead stain. So now we're just looking for live and dead cells and not cytotoxicity, well, cytolytic activity from effector cells. Calcein AM's overall hydrophobic nature allows it to readily traverse the lipid bilayer structure of the cell membrane in a typical concentration gradient-dependent manner. Once inside the cells, the hydrophobic and non-fluorescent Calcein AM is quickly hydrolyzed by intracellular esterases. This leads to cleavage removal of 2 nonpolar AM groups. Once the AM groups have been cleaved, the resulting polar and hydrophilic and now fluorescence-capable calcein dye is efficiently retained within the confines of the cell. Removal of the AM groups makes the cell-permeant probe no longer permeant. Now it's stuck inside the cell. Since dead cells possess insufficient amounts of esterases, they're unable to remove the AM groups. ICT offers a basic Calcein AM kit as well as an advanced Calcein AM kit. The first kit I'll discuss is the basic kit, which contains Calcein AM, a cellular assay buffer, as well as hooks for labeling DNA. To use Calcein AM, simply add the reagent directly to your cell sample, incubate, and then analyze. No wash steps are necessary. The calcein dye's inherently large quantum yield enables it to be readily detected within a wide variety of applications, such as fluorescence microscopy and flow cytometry, as well as a fluorescence plate reader. The degree of fluorescence correlates with the relative cell viability status. This slide I'm showing has some representative data that we generated using a fluorescence microscope. Those are the images shown on top, as well as flow cytometry results, which are the bottom histograms. The microscopy image shows live untreated Jurkat cells that were incubated with Calcein AM and then viewed using a Nikon Eclipse i90 microscope. The flow cytometry data shows live untreated cells as well as Jurkat cells that were killed by a 60-second exposure to 90% ethanol. The majority, roughly 93% of the live cells stained positive with Calcein AM, while none of the ethanol-killed cells stained positive. An overlay of the median fluorescence intensity generated from these 2 cell populations is shown over at the far right. ICT's advanced Calcein AM viability kit combines Calcein AM with 7AAD to allow for easy and simultaneous labeling of live membrane-compromised and dead cells within a single sample. Here, Calcein AM is used to detect live cells, which fluoresce green, while 7-AAD is used to detect the necrotic or late-stage apoptotic cells, which fluoresce red. Samples can then be analyzed using a flow cytometer or fluorescence microscope. The images shown here represent Jurkat cells that were either untreated, thus stained green, or were killed by a 60-second exposure to 90% ethanol and stained red. I will point out we do see a couple dead cells in our live cell population that stained red, but we do see no green cells staining in our dead ethanol-treated population. For the third panel I'm showing you, our live and dead cell populations were mixed together, stained, and then viewed using a fluorescence microscope. And as expected, since this is a mixture of the two cell populations, we see a mix of live green cells and dead red or membrane-compromised cells. Combining these two different types of fluorescent cell status indicator reagents within a single test allows for better resolution of the live and dead cell populations by making it possible to identify the percentage of cells that are 7-AAD positive versus 7-AAD negative within the green fluorescent calcein positive cell populations. Here we're showing some representative flow cytometry results. The majority of the live cell population stained green with calcein AM, and only a few cells stained red with 7-AAD. Meanwhile, the majority of the ethanol-killed cells stained red only. We don't see any green. As with the previous slide, I made a mixture of my live and dead populations and then evaluated that on the flow cytometer. And as expected, this mixture showed cells that were both live and stained green or were membrane compromised or dead and stained red. I would now like to switch gears and focus on our final topic, which is the process of autophagy. Autophagy is a conserved intracellular lysosomal degradation process during which cytosolic organelles and materials are enclosed within an isolation membrane to form an autophagosome. The outer membrane of the autophagosome fuses with the lysosome. The sequestered material is subsequently degraded within the autolysosome. Autophagy plays a critical role in maintaining homeostasis by preventing the accumulation of damaged organelles by disassembling unnecessary or dysfunctional cells and cellular components. Autophagy occurs at low levels in the cell under normal conditions and can be rapidly upregulated during times of starvation or stress, such as degradation activities. Such degradation activities serve to provide nutrients, such as amino acids, nucleotides, and fatty acids, as well as energy during periods of elevated bioenergetic demands. ICT's Autophagy Assay Red contains a proprietary cell-permeant aliphatic molecule that fluoresces brightly when inserted in the membranes of autophagosomes and autophagolysosomes. Our kit allows researchers to easily monitor autophagy using a flow cytometer that's equipped with a green-yellow laser and appropriate filter. The optimal excitation and emission for our kit is 590 and 620. This slide shows some results from an experiment where Jurkat cells were exposed to an autophagy-inducing or inhibiting agent and then analyzed by flow cytometry. What I'm showing you here is Jurkat cells that were treated with rapamycin, chloroquine, or a combination of rapamycin and chloroquine for 18 hours, and then stained, washed, and analyzed. For those not familiar, rapamycin functions as an inducer of autophagy, and it works by inhibiting mTOR, which mimics cellular starvation by blocking signals required for cell growth and proliferation. Chloroquine is an agent that prevents endosomal acidification. It accumulates inside the acidic parts of the cell, including endosomes and lysosomes. This accumulation leads to inhibition of lysosomal enzymes that require an acidic pH and prevents fusion of endosomes and lysosomes. Chloroquine inhibits autophagy as it raises the lysosomal pH, which leads to an inhibition of both fusion of autophagosome with lysosome and lysosomal protein degradation. Although chloroquine is known to inhibit autophagy, in the experimental model I'm showing you here, treatment with chloroquine actually results in an increase in signal since inhibiting fusion with the lysosome and subsequent lysosomal degradation actually results in a pileup of autophagolysosomes. So the combination of rapamycin and chloroquine causes the most significant increase in median fluorescence signal, as rapamycin induces an increase in the autophagosome formation, and chloroquine prevents the autophagic process from going to completion. So that is all I have for today. As you're planning out your project or working through an experiment, we do have a variety of resources available on our website to help you. You can find webinars such as this one on a wide variety of topics. Just go to the archive section on our webinars page. We also have some short product demonstration videos if you'd like to see some of our popular kits in action. We periodically update our blog with helpful tips, new citations, company announcements, and so on. So please check there for all of your ICT news updates. We also have extensive documentation in the forms of product manuals, safety data sheets, and certificates of analysis that are easily accessible from our products pages should you need them. One of our points of pride here at ICT is our excellent customer service and technical support. Being a small company, if you have a technical question, you can frequently speak with the inventor of the product to get your answers or to help with just troubleshooting your project. We also offer fast and affordable shipping, quickly sending out orders the next day. Our number one priority is helping our customers achieve their research goals. So lastly, as a special promotion for our webinar attendees, here's a discount code that will get you 20% off the order of any assay kit now through March 15th. And with that, I would be happy to take a couple questions.
Wonderful. Thank you so much, Christy. As she mentioned, please feel free to chat in your questions to me. Again, usually we get lots of questions here, so if we aren't able to get to yours, we'll follow up with you via email right after the webinar. But to get started, I see some questions coming in here. The first question someone is asking is one we get a lot, and someone's wondering, how sensitive is FLICA, Christy?
Okay, thank you. Yeah, you're right, Jackie. We do get that question a lot. So our FLICA products are very sensitive. For example, this reagent is sensitive enough that it can detect natural apoptosis that occurs in control cells, like your untreated cell population. Ballpark estimate is that anywhere between 2 to 8% of your cell population is going to be entering apoptosis at any given point in time. And if you look at our negative control or non-induced cell populations, we often see roughly that percent of cells staining positive with our FLICA products.
Wonderful. Thank you so much. We're also looking at, I'm not sure what specific kit we're referring to here, but we're looking at what types of instruments are compatible with the kits.
Sure. I realize that might get a little confusing. By and large, most of our products are compatible with a fluorescence plate reader, fluorescence microscope, or a flow cytometer. There are some exceptions. If you think of those cytotoxicity kits, where probably that would be really difficult to evaluate using a fluorescence plate reader given the 3 different fluorophores. Some of those are maybe just best evaluated using a flow cytometer. Definitely take a look at the product listings on our website to check out any particular product of interest and make sure it's compatible with your desired approach to monitoring your fluorescence output. We do have all that information listed on the product page.
Wonderful, thank you. We have a question here with regards specifically to the autophagy kit. What do you recommend for generating a positive control population for the autophagy kit?
That's a great question. There are many different chemicals out there for inducing or inhibiting autophagy. In the sample data I showed in this presentation, I evaluated cells that were treated with rapamycin and or chloroquine to induce autophagy. The best inducing agent will likely depend on your cell line of interest and ultimately on what you're investigating. So if you're unsure of where to start, you might consider browsing the list of autophagy-inducing agents or autophagy-inhibiting agents. A common place I go to look for agents is a company called Invivogen. They carry a wide variety of autophagy-inhibiting and inducing agents in case you're interested. Alternatively, it's been shown that starvation can induce autophagy. You could certainly try culturing your cells in something like an Earl's balanced salt solution for a couple hours to trigger this starvation to induce autophagy.
Wonderful, thank you. The next question we have here, are these products compatible with samples that have been fixed?
Okay, well that one depends on which product you're interested in. Regarding the FLICA-based kits, which as I've talked about before are things like our Apoptosis and Pyroptosis kits, as long as you can stain your cells with FLICA, then the kit's compatible. Just as a reminder, FLICA only binds to active Caspases. So this kit will not work with cells that were fixed prior to staining, as this process would inactivate any Caspases that were present. As a kind of way of reassuring yourself that you can fix your samples, I want to point out that ICT includes a formaldehyde-based fixative in kits that are compatible with the fixing process, as well as a protocol in our manual for fixing such samples. We do recommend that our customers avoid using any absolute ethanol or methanol-based fixatives, as these may inactivate some of the fluorescent signals from some of our products. Our Autophagy kit is also compatible with fixative. I do want to point out our Calcein AM kits are not compatible with fixative. In general, as I mentioned, if a kit's compatible with fixative, we do our best to make that clear in the product information on our website. Similarly, if a kit's not compatible with fixative, we also point that out on the product literature as well.
Sounds great. Thanks, Christy. We'll take just one more question here before we wrap up today. How many tests are included, and what constitutes one test for these kits?
Sure. I realize I covered a wide variety of different tests today, so that answer depends on which kit you're talking about. So most of our sample sizes for one test vary from somewhere either it's 0.3 milliliters or 300 microliters up to half a mil. For the most part, the FLICA-based kits were designed with a 0.3 mil sample size in mind. For some of our products, we do offer two different sizes. We offer a trial size or a standard size. The trial size typically contains enough reagent for roughly 25 to 50 tests, assuming that 0.3 mil test size, while the standard kit size typically contains enough reagent to stain between 100 and 200 tests. Overall, the number of samples that can be evaluated is a function of the staining concentration you use. And this can vary depending on the cell line or type. In general, the optimal staining concentration and staining duration, incubation period, should be determined experimentally. So overall, the number of tests and the number of tests that we recommend for any one kit, as well as the staining protocol provided with each kit, it really just reflects our general guidelines or recommended starting points. Each experimental situation might be a little bit unique and need to be fine-tuned for that exact purpose.
Thank you, Christy. Well, that's about all the time that we have for today. If we were unable to get your question or your question required a bit more of an in-depth technical response, we'll follow up with you personally after the webinar. Feel free, if you haven't chatted yours in, to chat in as we're signing off here. Again, I'll be sending out the webinar recording to everybody within the next day here. Our next webinar is scheduled for March 22nd and will be on the topic of inflammation, pyroptosis, and detecting caspase 1 activity. You can register for this particular webinar on our webinars page. And if you think of any other questions after the webinar or you have topic suggestions for future webinars, please feel free to contact us or fill out the Contact Us form on our website, or you can even reach us through social media on Facebook, LinkedIn, or Twitter. Thank you again, everyone, so much for joining us, and have a wonderful day.